Hello everyone. The topic of this lecture is cardiac septation. This is the first subsection of this lecture, which is about the methods of cardiac septation. This lecture has been further subdivided into two sections. That is the two methods of the cardiac septation. These methods are with the help of endocardial cushions and without the help of endocardial cushions. We'll discuss these methods in detail now. So, what are the methods of septation? So before getting into that, we should see the timeline when the septation begins to develop. Septation is actually a division between the common atria and the common ventricles. Initially, the heart tube has a single atrium, that is the common atrium, and the common ventricle. Here you can clearly see that in the figure. However, then a division tends to appear within the heart, and as the heart forms the adult heart, it forms two atria and two ventricles. This is the division that has been formed within the heart. So this is the septation. It tends to be formed between 27th to 37th day of development. Now, during this process, the embryo grows from 5 millimeter to 16 to 17 millimeters in length. That is about 3 times the length of the embryo in the beginning. So you can see how the size of the embryo is being enlarged at the same time as the septation is being made within the heart. So as we talked about earlier, there are two methods of septation, that is one with the endocardial cushions and one without the endocardial cushions. So we see actively growing masses which approach each other till they meet in the midline. So here you can see these masses. These are the ones in blue are the actively growing masses which are growing, and they tend to fuse with each other. When they meet with each other, they form the cardiac septa. This is the cardiac septum, which has been formed by adult growing masses, and now it has been divided. It has divided the atria and the ventricles into two parts. Now, another method is if a narrow strip may fail to grow. For example, this method occurs in the ventricles if a narrow strip of a muscle has failed to grow. Now the adjacent areas would continue to grow till they approach each other and they fuse with one another to form a septum. So here you can see it is not clearly evident in the picture, as that would be a faulty partition. The picture is always this picture depicts a complete true partition that has happened, but in the second method, a narrow strip may fail to undergo development, and as a result of which the remainder of the cells continue to develop so that they overcome this failed area of cell growth, and then they help in the formation of septa, resulting in the formation of two separate ventricles, as this process commonly takes place within the ventricle. So let's talk about the endocardial cushions. What are the endocardial cushions? Basically, endocardial cushions are actually proliferations of the extracellular matrix which are bound by the cells of the cardiac, bound by the cardiac myocytes. So there is increased synthesis of the extracellular matrix. Here you can see this is an endocardial cushion where there is increased synthesis of the extracellular matrix. This produces the endocardial cell-covered protrusions of extracellular matrix into the cardiac lumen. So as I mentioned earlier, endocardial cushions are basically protrusions of the extracellular matrix which are covered by the endocardial cells, and they are pushed into the lumen. So this is an endocardial cushion. This is actually a protrusion of extracellular matrix which is covered by the endocardial cells and is pushed into the lumen. So endocardial cushions, they help in the development of the atrioventricular region. This is the atrioventricular region. They help in the development of AV region. They help in the development of conotruncal regions. Conotruncal regions are the outflow tracts which are formed in approximately this region. So they help in the formation What is the function of endocardial cushion? They help in septation and formation of the atrioventricular region, that is the communication between the atria here and the ventricles here, and also the communication of ventricles to the outflow tracts, which are the aorta and the pulmonary trunk. So let's talk about endocardial cushions. So these are the endocardial cushion. This is the central endocardial cushion. They help in the formation of membranous atrial and ventricular septa. This is the atrial septum, and this is the membranous part of the ventricular septum. So they help in the formation of membranous part of the atrial as well as the ventricular septum. They help in the formation of atrioventricular canals, as we talked about earlier. These are the AV canals, and these are the endocardial cushions, which are helping in the formation of AV canals. AV canals are actually pathways between the atria and the ventricle through which the blood can flow. And they also help in the formation of pulmonary and the aortic channels. The pulmonary and the aortic channels are the outflow tracts of the ventricles. These are the tracts through which the blood exits the ventricles, and the endocardial cushions help in the formation of these outflow tracts. So what happens without endocardial cushion? It more commonly occurs in the ventricular septum, so we'll talk about it. A narrow strip of tissue may fail to grow, while areas on its side continue to grow. For example, a certain piece of tissue at the cursor of my pointer of my cursor may fail to grow here. If the tissue fails to grow, however, the tissue around it may continue to grow, as a result of which this growing tissue may overlap this failed tissue and the septum may be formed whatsoever. However, a narrow ridge is formed between the two expanding portions because the central portion is not undergoing proliferation. However, the side portions are continuously undergoing proliferation, so a narrow ridge might be formed between the two. So the growing portions approach each other and they eventually merge. Obviously, these growing portions which are continuously growing, though there's a ridge in between, but they'll keep on growing till the point they merge with each other. However, this does not completely divide the lumen, so it may not completely divide the lumen. So this was all about this section. In this section, we talked about various methods of septation that are seen in the heart. These methods were with endocardial cushions and without endocardial cushions. So in the method of endocardial cushion, we saw extracellular matrix protrusions which were covered by the endocardial cells. They went into the lumen and tend to divide and proliferate, and hence helped in the formation of the AV canals, the outflow canals, and the interventricular, the membranous portions of the atrial and the ventricular septa. We also saw the other method of septation which occurred in the ventricles, in which a portion failed to grow and the contiguous sides continued to grow till a ridge was formed. Now the proliferating cells kept on proliferating until they merged with each other. However, this partition was not complete. I hope you enjoyed this section. For further sections, keep watching sqadia.com.